Hello, I'm Charlie. I'm a first-year PhD student at the University of Lincoln. The title of this talk is basically just the overarching themes of my PhD, but I'm just going to be introducing katydids, talking about their communication systems, and basically just persuade you that they're really cool animals to study. So katydids, or bush crickets as they're also known, they belong to the family Tetygonidae in the suborder Ensiphera. I won't delve too much into taxonomy, but basically they're the sister group to the Calyphera, which is the grasshoppers and locusts, and they're closely related to field crickets, weta, and a few other groups. And now one of the features of these guys, which has interested ecologists and evolutionary biologists, is their mechanisms for communication, which are probably some of the most advanced and musical of all insects. So the way by which most katydids communicate is the process of tegmenal stridulation. This is the process of sound production by moving a hardened scraper on one wing against a row of teeth called the file on the other wing. The sounds produced by this process are almost always species specific, and they range in sound frequency from low-pitched calls like this species, Mecopoda elongata, which sings at around 6 kilohertz, and then we have the genus Supersonus, which can sing at over 150 kilohertz. We, on the other hand, are lucky if we can hear above 16 to 20. So katydids have been subject to a constant evolutionary pressure for the last however many million years to evolve ears capable of hearing all these calls. Not only that, but around 60 million years ago, they were faced with the emergence of a new type of predator, echolocating bats. And there's lots of evidence that katydid ultrasonics have actually evolved in part due to predation pressures by bats in the need to hear echolocation to avoid certain death. So there are many other ways in which katydid hearing has been under selective pressure, but basically it's resulted in the need to have a rather sophisticated hearing system. And as weird as it is, they have their ears in their front legs. So each leg possesses 2 eardrums or tympana, and they also have a hole called the acoustic spiracle, which is located just behind their front leg. And this collects sound and sends it to the ear through a specialized ear canal called the acoustic trachea. And these features are evolutionarily derived from the respiratory system. So with 2 eardrums in the front leg and an additional spiracle behind the front leg, and if you double that for both sides of the body, the katydid actually has 6 inputs for sound. Now this ear itself also displays an extreme variation in morphology. Many tympana are exposed, such as this one on the left, but others possess cuticular flaps or pinnae, and these are believed to assist in understanding the direction of sound. And also to increase ear sensitivity. And this is just a nice example showing you all the different variations in morphology that we observe. There's still no agreed theory on the function of these, but we know from some individuals who actually have these flaps on backwards that they've probably evolved several times independently. So how does this ear actually function? Well, in cross-section we can start to see this. So once the ear canal or the trachea reaches the ear in the front leg, it divides into an anterior and posterior branch. And essentially the 2 tympana vibrate in response to sound coming both through these tracheal branches as well as sound hitting the ear externally. So the resulting motion of the eardrums is just basically a pressure difference receiver mechanism receiving the difference between internal and external sound input. When these tympana vibrate, they're connected to a tympanal plate, which also vibrates, and this sends waves of pressure into a fluid-filled auditory vesicle. Now, this vesicle lies on perhaps the most important organ of hearing in these animals, and that's the crista acoustica, which is located just below the auditory vesicle. So this organ is a collection of linearly arranged mechanoreceptors. They perceive the motion of the fluid in the auditory vesicle. Depending on the frequency of sound the ear is receiving, this fluid peaks at different points along the auditory vesicle, and this activates different mechanoreceptors. So what this basically means is they organize the ear by frequency, with the high-pitched sounds at the distal end of the ear and the low-pitched sounds at the proximal end of the ear. So the katydid ear is, in essence, a mini piano. Now, I'm sure some of you may be aware that actually mammals possess a similar ear structure. So our inner ear cochlea is also arranged by frequency. The only difference here is that it's coiled instead of linear, but it still displays this frequency arrangement that we call a tonotopic distribution. And in fact, the katydid ear compares on several levels to the mammalian ear. They both share a tympanum for sound reception, an impedance conversion mechanism, and that's the inner ear ossicles in mammals and the tympanal plate I just mentioned in katydids. And then they have this liquid-based organ for frequency analysis. So of course, this makes katydids a great model for comparative approaches of understanding both this type of ear and the extent of convergent evolution between two ridiculously unrelated groups. Now, fortunately, one way in which evolution will be assisting us in this study, and one of the primary aspects of my project, is the fact that some katydids display ridiculously transparent cuticle, which appears to be, dare I say, designed for investigating hearing. In this newly discovered species, top left, the cuticle of the body is actually so transparent that we can view just under a normal light microscope. Sorry, the image in the middle here. These are the mechanoreceptors of the Crista acoustica without any manipulation of the animal. In addition, on here on the right, this is the acoustic trachea that sends sound through the leg, and that's again just viewed under a normal light microscope. Another species display varying levels of transparency, highlighted by the graphic in the bottom left. And this, of course, allows for completely non-invasive recording of the auditory vesicle fluid in an intact animal without causing any harm. We can record this fluid, which we know is associated with the mechanoreceptor activation, and infer what they're hearing. So here, for example, we see the inner ear, and you can see the tonotopic arrangement in that depending on which point you're looking at, the frequency of maximum response changes, increasing in frequency moving down the leg. Just for a few extra thoughts on this, I'm not going to go too much into the active mechanics, but essentially what I mean by active mechanics is whether the insects use their own physiological energy to enhance their hearing sensitivity. So in mammals, there's a system called the cochlear amplifier. This is a mechanism in which outer hair cells which receive sound undergo a conformational change in shape, and they actually amplify sound-induced displacement to send stronger signals to the inner hair cells for consequent neural processing. And this is a physiologically vulnerable process, but enhances the hearing by about 30 to 40 decibels. So far, evidence for ear amplification mechanisms have been found in tree crickets and mosquitoes as well, but there are very few invertebrates where it's actually been tested, so we're only really starting to understand this process. So the main part of my project will be investigating these indicators of whether they do drive energy into their hearing to be more sensitive. That's basically it, really. Flew through that, but basically, thank you. Just to introduce katydids, I hope you all agree they're really cool. And if you've got any questions, obviously ask now or send me a message later.
That was extremely interesting talk. I loved it. Before I open up for any questions, I have to ask you: are there any other animals that have ears on their legs or any other weird places? I need to know.
No, they're not on the legs, but actually, insects in general are quite interesting. So because of their tracheal system, which extends all throughout the body, and because they have proprioceptors, which are a type of mechanoreceptor that interact with the exterior body and respond to motion, basically they can evolve ears on almost any part of the body. So you see ears in the palps, on the face, loads on the abdomen, the genitalia, all sorts. So katydids are just one example of that.
Wow, I did not know this. Very interesting. Thank you. Does anyone else have any questions for Charlie they'd like to ask? I think there's something in the chat.
So yeah, I just had a quick question. So can you see the katydid as a model being useful for the study of any other related species in terms of conservation? Because I'm guessing that because they've got quite translucent legs anyway, as you said, it involves sort of minimal manipulation of the katydid itself. So I'm guessing that's sort of advantageous from the perspective that you don't have to, I guess, bring harm to the katydid itself to research it. So can you see it being useful for anything that's quite similar as well in terms of the similar research?
Yeah, definitely. I mean, their ears are certainly the most transparent parts, but katydids are probably an understudied group in that they have quite a few features that are either enhanced or unique that nobody really looks at in them. They've got, for example, they have some of the most complex reproductive systems, which obviously if you've got a transparent animal, you could use that to your advantage. But I think for these guys, the main part is the fact that this inner ear is so accessible in the transparent animals that is hopefully just going to replace the need to use mammals and other things in experiments.
Yeah, cool, thank you.
I think there's one on the chat. Could these be used as a more trackable model for mammalian hearing? From Sean.
Yeah, I think definitely. So the only thing we've got yet to prove really is whether these non-invasive recordings we're doing have any correlation with the sense of the actual neural responses. So we're going to have to do some invasive hook electrode experiments, or we're looking at doing calcium imaging as well. But if we can prove that they've got that similar structure, we know that the sound's being activated in that tonotopic arrangement, then basically we can make basic comparisons to the mallet, definitely.
Can I just ask what kind of calcium imaging you'll be doing, out of interest?
I've got absolutely no idea yet. Not a clue.
Fair enough, fair enough. Well, thank you very much. That was really interesting.